السلام عليكم ورحمة الله وبركاته. طلبتنا الأعزاء، محاضرتنا هذا اليوم المحاضرة الرابعة اللي تخص كلاس ساركودينا of amoebal protozoal parasite. حقيقة اليوم سنتناول two genus related to this group. The first genus is Amoeba pushelli. This parasite is an unpathogenic amoeba with worldwide distribution, although not as common as Entamoeba coli or Indolemax nana. It means this parasite can be found globally and all the worldwide, but its incidence, its prevalence, not too much as like that found with both species listed precisely, Entamoeba coli and Indolemax nana. The life cycle for this parasite is similar to that found or associated with the Entamoeba histolytica, but the difference here is there is no invasive stage, no invasion to other system or to other organ that can be circulated within the infected individual circulatory systems. Lymphatic or blood systems cannot transmit such protozoa to other organ, not metastasis. Other slide here can show you the morphological feature of the trophozoite stage. You know, the life cycle is similar to that found with the Entamoeba histolytica. It has two stages in its life cycle. First, trophozoite stage, motile, and protective stage, which is cyst stage. Trophozoite stage of this parasite are actively motile, and on a permanently stained fecal smear, a nucleus with a large karyosome is evident. A large karyosome be evident, parent, and chromatin bodies form striations around the karyosome. This large karyosome surrounded by chromatin bodies that form striation around it. So its unique or specific criteria represent such stage of this protozoa. And the cytoplasm appears granular, containing vacuoles with ingested bacteria and other debris, food, bacteria, and other. Now, in this stage, here two pictures on the left represent the trophozoite stage of Amoeba pushelli, and the other side represent nucleus of Amoeba pushelli. The trophozoite stage here having one nucleus and its position not central, eccentric, especially in the mature trophozoite stage, as shown here in this picture. This nucleus not have peripheral chromatin. Its nuclear membrane, the nucleus of this stage, doesn't have peripheral chromatin, just nuclear membrane, and inside it there is a large karyosome, usually posited at the center, usually central position. Now, the chromatic granules surround the karyosome to form striation shape or phenomena. And within the cytoplasm there is vacuoles, many types of vacuoles, contractile, food vacuolae. Some of these can ingest bacteria, one be food vacuolae, the other. Now we return back to its class. In the right we can see large karyosome, the chromatic grounds can form striation around the nucleus. It is so apparently here in this picture. Now other slide can show the characteristic features related to its cyst stages. Now cyst of Odomini bacilli have one nucleus, not 4, not 8, not 2, just one nucleus. In mature cysts, usually eccentrically placed. Here, when the cyst will be matured, the place of nucleus be eccentrically placed or deposited, and chromatoid bodies are not present. Chromatoid bodies are not present, and glycogen is present. Glycogen mass is present as a compact, well-defined mass, staining dark brown with iodine. Here, this the main criteria of the cyst stage. Having one nucleus present eccentrically, there is no chromatoid bodies. Glycogen is a compact, large, well-defined mass, staining dark brown with iodine. Now, this slide can show you three shapes to the cyst stage of Odomini bacilli. At the left, schematic representation for this stage, and in the middle is stained, and in the right is stained also. But at the schematic shape we can see granular cytoplasm, large glycogen vacuole, there is cyst wall, and eccentric karyosome, and there is no nuclear membrane. There is no peripheral chromatin, whereas the chromatic grounds can surround the karyosome to form striation shape and or form, and no peripheral chromatin as shown in this slide. Now this slide can show the laboratory diagnosis for this parasite, and it is made by finding the characteristic cyst stages in an iodine stained smears or formal ether concentration method. Indeed, the trophozoite stage are difficult to detect in a wet preparation. Trophozoite stages are difficult to be detected in a wet preparation. These criteria of this concern, how can we diagnose this parasite? Now, under microscopic field, this permits detection of motile trophozoites of Odomini bacilli and can also provide information on the content of the stool. Example, the presence of leukocytes, RBC, fat, mucus, pus cells, crystals, other debris. Now, Actually, any case to be detected in laboratory, any case of disease, any situation, any disease, when you want to diagnose it precisely, you must depend on the after clinical symptom determination of clinical symptoms of the targeted patient. You must form macro examination before micro examination. Why? Because from macro examination you can detect and elucidate and show the color of the specimens, the odor of the specimens, the content of the specimens, blood, fat, mucus, and other. Solidity of the specimens also can be viewed. So there is bloody diarrhea, for example, fatty diarrhea, starter diarrhea, mucus diarrhea, and so on. So any specimen should be examined macroscopically, then followed by microscopic examination. Okay, the second species here in this lecture is Blastocystis hominis. Blastocystis hominis is also having certain criteria causing its identity. So, it is an inhabitant of the human intestinal tract. I mean, it's found in our GI if we affected by this parasite. And previously regarded as an unpathogenic yeast in the past, when we discovered it from the scientists and the researcher, and the study showed that it is an unpathogenic but related to a yeast, fungal species yeast. But nowadays and recently it be regarded as amoebic protozoal parasite. Also it is non-pathogenic, but it is puzzle in this situation, in this case. Now, its pathogenicity remain controversial. So there is a problematic in its pathogenicity. And the organism is found in stool specimen from asymptomatic people as well as from people with persistent diarrhea. The following step may be give conclusion for why we regard it non-pathogenic or its pathogenicity remain controversial or with puzzle or problematic issue. And Blastocystis hominis parasite is capable of pseudopodia extension and retraction, both extension and forward and backward, and retraction, and it can be reproduced by binary fission or sporulation to type. Other criteria: the classic form that is usually seen in the human stool specimens varies tremendously in size, not take unique size or limited size, but ranged from 6 to 14 micrometer. It is very, very wide length of variation in its size, from 6 to 40. It is long, different. And other criteria here is the cyst won't be occupied or enclosed within the thin walled cyst. If the cyst having thin wall, I mean cell wall, the cyst can cause auto-infection at the same individual, are the same affected guys or humans. But when the cyst be walled by thick wall, it can be responsible for external transmission through fecal oral route. You know, fecal oral route transmission can be related to other species of amoeba group. Now, indeed, and actually, the presence of such a large numbers of these parasites, Blastus hominis, the number be 5 or more per oil imaging microscopic field. And the absence of other intestinal pathogens indicate the disease. So if it's found in a high number, for example 5 or more, and we when examined it under oil imaging lens in our microscopic field, and we can see the absence of other intestinal pathogen, thus will indicate account for other disease or disease. So the organism may be detected in a wet mount or trichome stained smear of fecal specimens, and the treatment or its treatment can be accomplished with odocanol or metronidazole, has been successful eradicating the organisms from intestine and alleviating symptoms. Now, however, the definitive role of Blastus hominis parasite in disease remain to be demonstrated. It is still puzzled, is still understood well, remain to be demonstrated. The incidence and apparent worldwide distribution of the infection by this microbe or by this protozoal parasite Blastus hominis indicate preventive measures to be taken, which involve improving personal hygiene and sanitary condition or sanitation requirements must be applied to prevent, to eradicate, to limit such exposure by such protozoal parasite. As you know, most infection can occur by various microbes, pathogenic microbes, by the defect, as a result from the defect in the health level, especially personal hygiene and sanitation condition, and maybe related to individual behaves, and other reasons can bring about to impede to outbreak of such microbes or protozoal parasites. Now lecture is ended, and I thank you all, my students, for your listening, and I wish the lecture is be simplified and be easable to be understood, and any question, any explanation you need from me, I can furnish your mind. And again, thank you, my student.